Hello, my name is Jency Rajan. I'm a final year PhD student within the Personalised Medicine Centre at Ulster University in Northern Ireland, UK. My poster is on my PhD project and the title is An AI Approach to Identifying Novel Therapeutics for Rheumatoid Arthritis. The hypothesis of my project is a systems approach can be used to predict a combination of therapeutic options for the treatment of rheumatoid arthritis to achieve greater efficacy with minimized toxicity. I have three main aims. Number one, develop, test, and refine a connectivity mapping-based bioinformatic pipeline called Drug Express, which has the capability of mining treatment response gene signatures. Two, verify treatment response signatures and prioritize novel drugs to generate a list of robust sensitizing drugs. And three, validate the effects of novel drugs in an in vitro model system of RA. First, to give a bit of background, rheumatoid arthritis is a systemic and chronic autoimmune disorder that affects musculoskeletal joints. It has a global prevalence rate of 0.24 to 1% and an incidence rate of 1.5 and 3.6 per 100,000 in men and women in the UK. This condition has significant impact on quality of life and work capacity. There is 15 to 46% increased mortality compared to the general population. Around 1 in 6 people have major depressive disorder associated with increased levels of pain and functional disability. 30 to 50% of people within the first year of diagnosis have to remove themselves from the workplace, and approximately 68% of patients in the UK are physically inactive. This becomes a vicious cycle of disease progression and increased pain, thus affecting both their physical and mental health. Currently, treatment and management of RA is aimed at gaining control of inflammation and reducing pain. However, achieving remission or low disease activity with minimal toxicity isn't possible with the current suite of drugs. This figure here illustrates the mechanism of action of the main drugs in each drug class used for treatment today, and it shows the impact it has on the pathology. With escalating de novo drug development costs, bioinformatic discovery pipelines offer the ability to repurpose already licensed drugs and explore synergistic combinations more efficiently. Moving on to methods. This figure here shows the Drug Express pipeline that was developed. It is an in silico drug discovery and repurposing pipeline with an in vitro validation endpoint. This can actually be divided into 4 different steps. Number 1, identification of RA datasets. Number 2, identification of differentially expressed genes. 3, connectivity mapping to identify candidate drugs. And number 4, validate in model systems of RA. The results section can also be divided into 3 parts according to the methods. Number 1, identification of RA datasets. For this, public databases such as GEO and RA Express were mined to gather a collection of 27 suitable datasets for data analysis. These datasets were filtered, sorted, and selected using a predetermined criteria. Datasets shown in this table were identified as being suitable as they contained post-treatment gene expression profiles from patients treated with a variety of drugs, and it also contained information on whether they were responders or non-responders to that treatment. 2. Identification of differentially expressed genes. Differential expression analysis was conducted comparing responders and non-responders, and differentially expressed genes were identified. Volcano plots on this slide shows the differentially expressed genes labeled from individual datasets. Significant genes were then identified based on a cutoff criteria of an adjusted p-value of less than 0.05. Significant genes from individual datasets were then merged and mapped to Affymetrix probe set IDs and used to derive gene signatures characteristic of treatment response. 3. Connectivity mapping to identify candidate drugs. Gene signatures were then plugged into a connectivity mapping platform called SSE-MAP. After analysis, a detailed table of results and a volcano plot is produced as shown here. The x-axis is the connection score of each compound in the connectivity map, y-axis is the log p-value, and the green line is the significance threshold. A total of 7 compounds were identified as being statistically significant. Candidate compounds were then selected based on a cutoff criteria and whether these enhanced or suppressed the response phenotype. A total of 6 statistically significant candidate drugs with high probability of inducing therapeutic response was identified. The next step is to perform in silico toxicity screening on the list of candidate compounds to focus in vitro tests on a list of optimal compounds. Benefits of this approach include the identification of novel drug combinations and dosage regimens. It can be applied to a new index disease and can be used to identify established treatments as priority candidates for further validation. Challenges include the availability of large, reliable data warehouses and high-quality datasets, the varying standards for annotation, formatting, storage, and access, and another challenge is dealing with the data noise generated from incorporating multiple different datasets. In conclusion, analysis with the Drug Express pipeline illustrates the potential of extracting significant genes characteristic of treatment response from public expression datasets to predict new drugs which may offer new treatment options to refractory patients. The efficacy of novel drugs discovered to reduce gene expression and cell proliferation will be screened in an in vitro fibroblast model ahead of preclinical studies. Thank you so much for listening. I'd like to take this opportunity to acknowledge and thank the Department of Economy and the Northern Ireland Rheumatism Trust for funding my PhD project, and also my supervisory team, Dr. Shudong Zhang, Dr. David Gibson, and Professor Tony Beerson for all their support and guidance. Thank you.